Hello everyone, we're going to look today at just the remainder of what we didn't do in class the other day. This is the second part of Chapter 4, Adaptive Immunity. And again, to make sure, just to be explicit about this discussion, what we're talking today about is how B cells and the resulting antibodies that are produced are the humoral immune response, which is part of the adaptive immune system. Humoral immunity occurs when the body is breached by foreign pathogens that have gotten past the non-specific immune response, those physical and chemical barriers, and the host is exposed to materials we call antigen, bacteria, viruses, pollen, allergens, any of those kinds of things, parasites, and this results in an activation of B cells that produce antibody. This slide is just a quick review of what we saw the other day, and we're looking at this portion of it, the B cells. B cells are the lymphocytes that remain in the bursa equivalent of the bone marrow that are going to differentiate into B cells. So as bone marrow, pluripotential or hematopoietic stem cells develop into the common lymphoid precursor. Whoops, doesn't like me when I'm using the arrow. Develop into B cells, and remember, in the bone marrow we can also look at natural killer cells, but we're not talking about that today. Why? That's specific immunity, right? The B cells, as I said, are involved in the production of antibody. As I said, humoral immunity really results in, our goal is the production of antibody. B cell differentiation starts in the bone marrow from that hematopoietic stem cell. Going to develop into the common lymphoid precursor and move on to what's called pro-B cells, pre-B cells, immature B cells, which it then leaves the bone marrow and develops and further differentiates out in secondary lymph tissue into mature B cells. The hallmark of the B cell differentiation cycle is to develop not just B cells, but immunocompetent B cells. There are going to be a number of stopgap measures along the way that are going to be little tests of all of the B cells that are being produced to make sure that they're going to be able to have an ultimate goal of producing antibody in plasma cell, as in the form of a plasma cell. We're going to see that B cells are activated by the presence of that foreign antigen. So this is going to start, B cell differentiation is going to start as a antigen independent cycle where they're going to start to develop before they've ever been challenged by an antigen, but by the time we get to the end, they're going to be antigen dependent because the cells are going to need to be exposed to a specific antigen in order to produce antibody. We are looking at the figure to the right here, and it depends on how big your screen is on your computer. If you want to, you can look at page 51. Yep, 51 of the book, and follow along. So what we're looking at in the figure to the right is the differentiation of B cells, and this is where we're going to start. We start with the hematopoietic stem cell in the bone marrow. And as I said, this is the antigen independent phase. Doesn't matter what we're going to be producing, what this B cell may ultimately be come in contact with antigenically, because in the beginning we're just developing and maturing B cells. The stem cells differentiate to pro-B cells in the stroma, or the bone marrow stromal cell. Within the stroma, there are stromal cells within the bone marrow, and actually other organs have stroma as well. But the stroma of the bone marrow is really just kind of the connective tissue cells. The stromal cells are the connective tissue of the bone marrow, and the purpose of them really is to form kind of a support system for the bone marrow stem cells to develop and differentiate further. And what happens is within the stroma, there are cytokines, in this case growth factors, that will aid in the process of further differentiation. So the first step in the differentiation is this pro-B cell. And as the pro-B cell differentiates with these growth factors, what happens is, the genes of the pro-B cell that will ultimately be responsible for antibody production are rearranged with the design of being able to code for something called the heavy chains of the immunoglobulins. We're going to be talking about immunoglobulins and antibodies, same thing, right? In chapter 5 in kind of great detail, but we're going to look at it quickly today. Just to be clear, before we move on to the structure of the immunoglobulin that you see in the picture here, only when there's been successful rearrangement of those genes that are going to produce some of the chains called the heavy chains will the pro-B move on to the next stage called the pre-B status. If they're incapable of producing these heavy chains, the pro-B cell will be deleted. So we'll go through what we're talking about, and then I'll go just reiterate those 2 points one more time about how it's going to differentiate to pre-B cells. So because we haven't spent a lot of time on antibodies yet, it's next chapter, I wanted to give you a quick tutorial on this. This isn't in your slide deck because I actually decided to put it in once I got to this point. So as I said, the differentiation of pro-B cells, the purpose is this genetic rearrangement of the coding for the chains that make up the antibody production. So antibodies, remember I showed you in a picture or an illustration on the board how they're usually shown to us in the shape of a Y. Conformationally in real life, they're a 3D structure that we'll go over, but this is the kind of the schematic drawing of an antibody. And they're characterized by every antibody has 2 heavy chains, these long ones right here, that's a heavy chain, and this is a heavy chain right here, and 2 light chains, bing, bing. The heavy chains are always the same. In this case that we're looking at in the genetic rearrangement, we're looking to be able to create something called mu heavy chains. We're going to look to see that the genetic rearrangement of these pro-Bs will be such that ultimately the next stage is going to be able to form mu proteins, mu chains. And so these heavy chains, these long chains, and these light chains are different. So these are the mu chains, and these light chains are just smaller chains. When we get to the pro-B stage, they're not going to be in their final form. We'll talk about that in a few minutes. But these, I guess the only other thing I would say about this, a few other things that we'll talk about this. So as I said, the heavy chains are always identical. Our first one we're going to talk about are the mu chains from the IgM, mu for M, antibody. The light chains can be the same or different. They can have different protein structures. So we're not going to spend a lot of time on that right now. We'll talk about that in chapter 5. The other important piece of this structure is right here. This is called the variable region. And much like the T cell, when we talked about it last week, about that genetic rearrangement of T cells during their development, and how we have those different versions of gene rearrangement in order to create antigenic diversity, the ability of structures that are formed within the cells to be in different formats and to recognize different antigens, diverse antigens. That's what this variable region is. So pro-B cells are really developing all different genetic rearrangements that these ends, these little tips, these variable regions are different. So that no matter what antigen it comes in contact with, hopefully it's able to mount an immune response to it. Let's see. And the last thing I would say here is light chains, just as a reminder of what they are. Light chains are short, heavy chains are very long with a little hinge right here that makes it bend. And they're so named, ironically, or not so much. Heavy chains are just heavier than the light chains. They're much, much larger, a little bit more than double in weight. And so the molecular weight of the heavy chain is heavier than the light chain. So the light chains have a lower molecular weight. The heavy chains have a higher molecular weight. So back to the point of the B cell differentiation and kind of its end stage. When the genetic rearrangement of the pro-B has been developed enough that the pro-B is able to then code for the production of heavy chains and ultimately light chains of antibody molecules, only when that happens will it move on to the next stage of a pre-B cell. If it's determined, if the body and that check and balance, that those stopgap measures, making sure that the cell is an effective cell. If it's determined that the pro-B cell that's developing is not going to be capable of coding for the production, let's talk first about the mu chains, then the cell dies. The cell is deleted, and it's not going to go further in the differentiation stage. The pro-B stage, the pro-B stage, as I said, is the one where heavy chain, the genetic rearrangement occurs so that we have identified that heavy chains will be able to be produced. The pre-B cell, on the other hand, is no longer kind of in the theoretical. Once those heavy chain genes have been rearranged, those rearranged genes that were developed in the pro-B cell start to actually get transcribed within the B cells, the pre-B cells. That's hard to say 3 times fast. Are actually transcribed within the pre-B cell stage in order to actually synthesize these heavy chains. So these heavy chains start to be synthesized. And if you look at, pardon me, I'm going to go back up a page. You'll see in the cytoplasm, there are the little mu chains that I talked about. These are the mu chains that ultimately are going to aggregate and form antibodies. And right here, these are the light chains that are forming up here. This is an alpha chain and a beta chain. You can see these are 2 different chains. Remember, I told you that heavy chains are always identical. So in the pre-B stage, these antibodies that are developing are an alpha chain and a beta chain and 2 mu chains. The mu chains of the heavy mu chains will develop into what's called an IgM class. That's a really big, monstrous immunoglobulin that actually has 5 of these structures within one large molecule. We'll see that next week. The heavy mu chains are accumulating in the cytoplasm. We just saw this here. There they are, accumulating in the cytoplasm, and they're also membrane bound. Ultimately, they can express on the cell surface with what's called the surrogate light chains. And what the surrogate light chains are, remember I said that in the pre-B stage, there's our mu chain, and this is an alpha chain, and there's a beta chain. Those alpha and beta light chains are not going to be the final version of a light chain that gets produced from plasma cells at the end of this process. These are surrogate light chains. What it's telling us is that really the cell is capable of producing immunoglobulins. It's capable of producing light chains. It's capable of producing the required heavy chains that are needed. So once it gets to that point, this cell is getting to be in pretty good shape. So you'll hear that called the surrogate light chains. The alpha and beta are surrogate light chains. And these act as a signal in conjunction with another step that I'm going to talk about, I think, I will not talk about it on the next slide. I'll talk about it right here. So the pro-B cell had passed did the heavy chain rearrangement thing work to move it into the pre-B stage? The pre-B stage started synthesizing heavy chains. The surrogate light chains are there. And that success of production of the heavy chains and the light chains within the B cell, the pre-B cell, results in the formation of what's called a pre-B receptor. And the development of the pre-B receptor has to be met before these pre-B cells can move forward. So what did we have for these B cells to move forward? The cells with heavy chains, surrogate light chains, and that pre-B cell receptor. If pre-B, can I say this 3 times fast? If pre-B cells survive with a pre-B cell receptor, then what happens is those pre-B cells start to clone themselves. And that cloning process is called clonal expansion. So we're just going to start cloning cells as we go forward. And that is then termed, that cloning stage is called the immature B cell stage. So when it gets to the point of having met the requirements of being moving past the deletion of ineffective pre-B cells, you've got B chain, you've got heavy chains, you've got light chains, you've got the pre-B cell receptor formation. That signals the ability for clonal expansion, and you then start calling them immature B cells. Immature B cells are characterized by having a complete IgM on the surface of the cell. So no longer are the mu chains in the cytoplasm and the alpha chains on the surface, but you've got actual complete immunoglobulins that are bound to the surface of the B cell. The IG, and it's going to be IgM because why? That's a mu chain, right? The mu chain always represents, the mu heavy chain always represents IgM molecules. So they have got the IgM bound to the cell surface. So if you had a cell surface here, you'd have a little IgM sticking on, or many of them. You'd have many little antibodies here. And what they're looking for is to have the antigen bind right here. The specific antigen is going to bind to this as they start to develop. We talked in the past, though, that just because we've got some really important protein on the surface, like IgM, it doesn't mean that's the only thing that's on the surface, right? We talked about these CD markers, these protein markers, that can have a number of functions, the surface proteins. Sometimes they identify, they used in our, they develop like CD4 and CD8 on specific cells, right? So CD4 we know are on T helper cells, CD8 are on the cytotoxic T cells. There can also be CD markers, protein structures on the surface of a cell, and they can have other functions. So in this case, on the immature B cell, we also have what's called CD21 and CD40. And before I look back at the other picture, I want to make sure before I start slamming through, they don't show it on here. All they do is show on the page 51. All they show on letter C, the immature B cell, is that it's got a surface-bound IgM. So that's okay. But there are other proteins on the surface. One is called CD21, and one is CD40, and then we have class 2 MHC molecules. How come? Why do we have class 2 molecules on B cells? Well, we know B cells are responsible for what? Those pathogens that are brought into the cell, but they're multiplying and living outside of the cell normally. So they're exogenous antigens. They have to be brought into the cell, and those we need class 2 molecules for. So that's why we have class 2 molecules on immature B cells. But the CD21 has a purpose that we're going to see in a few minutes, that will promote the contact between B cells and antigens. We're going to talk about cytokines in another chapter. I can't remember where it is, 15 maybe? I've lost track. But CD21 really is promoting contact between B cells and antigens. It's going to facilitate that contact. And then CD40 in the class 2 MHCs, we're going to see in just a few minutes, is going to be important in the interaction and the binding of B cells with T cells, when they've both kind of come in contact with the same antigen. And how are those T helper cells going to help the B cells start to produce antibodies? How are they going to develop into plasma cells? This, you can actually ignore. This was a picture that I put in later on. So this is not actually a hyperlink that's going to help you. I am going to say, though, I'm going to apologize in advance. This is the reference for one of the pictures on the slide deck further. So you'll see this as we go further, and I don't think I put it in the references. I'll correct it the next time I update the PowerPoint, but this is a reference for one of the slides and one of the illustrations in the slide deck. So we're at the stage of immature B cells. It's at this point that the B cells are again tested. So we tested them earlier in, are they able to produce, do they make a sufficient genetic rearrangement in order to someday be able to produce the chains, the mu chains and the light chains, right? They passed that test. The next test, they checked, did they actually make antibody? Did they make the B cell, the pre-B cell receptor? So they passed that checkpoint. If they didn't meet those criteria, those cells died, right? Here's another point of a self-check. And in this case, the B cells, the immature B cells, are checked, just like we did for T cells, that they don't react with self antigens. If they react with self, those immature B cells are eliminated. They're inactivated and or eliminated in the bone marrow. And you can see here, 90% of the B cells die this way. Very much like the T cells, we're getting rid of all of these things that interact with self. And so this is a huge way that the number of B cells die off in the host. The process of that self-check, or I shouldn't use the word self, that process of checks and balances that identifies whether a B cell is reactive to self is a process called central tolerance. It's in red on your slide deck here. We're going to spend a lot of time on this process in autoimmunity. I think that's chapter 15, maybe not cytokines. I think this is chapter 15. But central tolerance, we're going to spend time and look at that in really kind of excruciating detail later. But for the time being, let's just remind ourselves that central tolerance is just the process of eliminating B cells that react with self. We don't want to produce antibody against ourselves. We end up with autoimmune disease if we do. So we want to get rid of those B cells that react with self. All of the B cells that survive this process of central tolerance review, those are going to go and they're going to move out to the spleen. We know that, let's see, When they move into the spleen, immature B cells, this is where they're going to develop into mature B cells. Up until this point, they were still in the bone marrow, right? They leave the bone marrow, they go into the spleen. We're not sending cells out into the spleen that are going to identify with self. So that was our kind of our last check there, our last check and balance. So we had really 3 good solid stopping points, maybe even 4 if you want to break out the pre-B cell issues into 2. But we had multiple steps where we could identify an immunocompetent B cell. When the immature B cells move out into the spleen, we know from our discussion of the secondary lymphoid tissue kind of what the purpose was. The cells that move out into the, not just the spleen, but really the secondary lymphoid tissues are known as either marginal B cells or follicular B cells. The marginal B cells, the ones in the spleen, we know kind of stay in the white pulp of the spleen, where they're really going to be a first responder to antigens that are in the blood and filtered in through the spleen by maintaining a strong and antigenically diverse B cell population there. Ones that can have a great deal of antigenic diversity, able to interact with a number of different kinds of antigens. It's going to be an effective immune response, I guess is all I need to say. As the blood filters through the spleen, they come in contact with the now mature B lymphocytes. They're going to be taken into the B lymphocytes. They're going to have peptides. We have the antigen broken into peptides, brought up to the surface on the MHC, and they're going to be able to be identified, and we'll develop some memory around that antigen, as well as producing specific antibody. So we're going to see that. So that's the marginal B cells. They remain in the spleen, stay in the spleen until needed, and then are allowed to quickly respond by producing IgM antibodies initially, and later on some more specific antibodies over time. So these are quick responders, relatively quick. It's not that fast, but relatively quick. The next are the ones that leave the bone marrow and go out to some of the other secondary organs of the the secondary lymphatic system organs, gut-associated tissues, things like Peyer's patches, and I call it the not-so-useless appendix. We always hear the appendix is not a useful organ any longer, but in the immune system, it's actually a place to house follicular B cells, where they'll also do some surveying for antigens, and they're allowed to circulate through the lymph system rather than through the blood. So that's the purpose of these mature B cells, where they are. They've left the bone marrow and they've gone out to places where they're going to find an awful lot of antigenic material, right? What else do we know about mature B cells? They contain both IgM and IgD. IgM, we know, is that first responder. I'm looking to see. Oh, let me talk about it here. I think I mentioned before it has really 10 antigen binding sites on it. It's a very, very large molecule, and it's a first responder kind of a molecule. IgD, I like to sort of call the I don't know. There's a lot of question about what IgD does in the immune response. We don't see it in kind of that direct antigen antibody process in our routine world, but we do know some of the functionality of IgD. IgD is a B cell activator. It's one of the ones, its presence activates B cells to when it's come in contact with an antigen. So IgD does serve a purpose in that it is one of the surface immunoglobulins that activates B cells when it comes in contact with an antigen. It also is known known weakly, that it helps to prolong the lifespan of those mature B cells when the B cell has been stimulated. So, what else do we know about mature B cells? Once they're stimulated, good point. Remember, up to this point, we had talked about starting in the bone marrow, we were in an antigen-independent phase. That whole development in the bone marrow and even release to the secondary lymphatic organs, it was still antigen-independent because it didn't require any antigen to develop the cell maturation and differentiation. Once we stimulate it with coming in contact with an antigen, we enter what's called the antigen-dependent phase of the humoral immune response. And that is really, you come in contact with an antigen at this point, and two outcomes are primarily going to happen. We're going to produce further differentiation of mature B cells into plasma cells, and those are the ones that are going to actually secrete antibody. And then we're going to produce some long-lived memory cells. Those are going to live, 6+ weeks and continue to be on the lookout for antigen within the host that matches their specific antibody, what they've been primed to look for. So let's look on. If we look at this side-by-side comparison of the actions of B and T cells relative to sort of a process that's going on at the same time. Let's start with the super familiar, which is what we've been talking about since the beginning, and that's the activation of the T helper cell. So here's our phagocyte, dendritic cell, macrophage, neutrophil, some kind of antigen presenting cell. And, in this case, remember, we're talking primarily about things like bacteria. So let's pretend it's a bacteria. The antigen presenting cell way up here comes in contact with something foreign. A bacteria, for example, takes it in, it engulfs it, it breaks it down into small peptides. The peptides get within the endothelial reticulum. I can't say the endothelium reticulum. Binds to the MHC class II molecule, and it brings it up to the surface to present it to what? A T helper cell. So there's the antigen, there's the MHC class II, and here's, there's the CD4 because this is a T helper cell, right? And there's the T cell receptor. So there's the MHC of or the binding site, the TCR, the T cell receptor, right here. I'm sorry, that's the CD receptor. I apologize. Right there is the T cell receptor with the antigen binding site to it. So this stimulates this T cell to this once naive T cell or virgin T cell. Now, once the T cell receptor binds to the antigen that's bound to the MHC of this antigen presenting cell, what does this T cell do? It clones itself, clones it, and says, I need to remember this antigen, and I need to clone myself so that I'm building some memory, right? This isn't addressing really the cytokine production and the cytotoxic T cell piece. We're really looking at developing B cell production, right? So the second thing is, I imagine you could say this is maybe the cytotoxic T, but then the T helper cell is also going to bind. Remember I told you that CD40 was important? So here's the CD40, which is helping to bind with the MHCs to the B cell that has also been activated by that. There's that antigen. There's that antigen. They're the same antigen, right? This B cell has been out here and it had engulfed it as well, produced an IgM antibody that is tagged with this antigen, and now these two, their CD40 binds them up together so that they are now working in concert. The T cell is helping this B cell do other functions. Remember, T cells are mediators, they're helpers. They're going to help something else happen. So in this case, by binding the B cell that's found that antigen and processed it and been producing a first stage of an antibody, the T cell is going to help it by giving it some additional cytokines, in this case an interleukin-2, that's going to cause proliferation and further differentiation. So let's look at the two stages of B cell production, right? We know that this mature B cell is going to proliferate, and it's going to produce what's called plasma cells, right? These are the antibody producing cells. And oh, here, look what this plasma cell is producing. Oh, those big old IgMs. These IgMs are going to go out, they're going to circulate, and they're going to look for more antigen. The antigen's going to bind off here. The complexes of antigen, once they bind off to antigen and antibody, once those complexes form, if you put a little, oh, there's my thing, you put a little antigen onto this antibody that's been in circulation, it actually is going to trigger another piece of the immune response that will end up, and we'll talk about it later. It's called complement, and it's going to trigger that response and kill that antigen, and then clear everything out of the body, out of the host. So that's a good thing. It's going to cause that little thing to lyse, that antigen to lyse. So that's the first stage of the proliferation, producing a plasma cell. Oh, we're going to produce some antibodies. What else does that plasma cell do? It releases additional cytokines that causes further differentiation of this plasma. This IgM, I told you, is a big old molecule. It kind of acts on that first response to an antigen by brute force. There's a lot of binding sites that aren't really a very strong lock and key fit with the antigen. They're not a great match to the antigen. They haven't defined themselves as being super specific to that antigen. They're clearly specific to that antigen, but they're not a great fit. Remember, I talked about kind of a key that you have to wiggle a little to get eventually the lock to open. That's an IgM. So you got to wiggle it a little, but it's also kind of like you just force it open. Same thing. So the IgM, because it's such a big molecule and there's so much of it, it forces the process to work. But the plasma cell wants to get better, and it produces a better antibody later. It's a slower response because it has to refine its identification of the antigen. Does more processing. The body is better to recognize. The more times it sees the antigen, the better the recognition of that antigen is. So ultimately, the plasma cells not only produce IgM, they produce other things. And in the case of a bacteria, we produce something called IgG. It's a smaller molecule, but it's super effective against the antigen. So this is the process of proliferation and the production of antibody. And then what else happens? We have additional, so this is the differentiation, right? Proliferation and differentiation produce this new plasma cell, developed from a mature B cell, produce the plasma cell, produce an antibody. Fantastic. What else do we know the second part of the humoral immune response is? Yeah, produce some nice memory cells that have this antibody on it. And this is the antibody that was specific to this little antigen. It's got it on the cell surface. And those memory cells are just waiting to come in contact with something else. And because we've developed these nice memory cells of this antigen, if it comes in contact with it, it doesn't have to go through this whole stage again. It's just going to bind an antigen right here and can go to generating some antibody. So that's the good news. These memory cells, remember we said they have a nice long life, 6 weeks plus in the host. But remember, they're constantly reproducing, cloning, and making more copies of themselves. So they have a nice long life on these, waiting to see if they encounter, some antigen to come back to see them. So this is that T cell piece, right? This is that antigen presenting T helper cell all the way to the production of antibodies and memory cells. So that's one phase. That's the action of T cells relative to humoral immunity. What about the B cells that just encounter the antigen themselves? So let's pretend that we've had that B cell that is triggered. You remember that we have B cells that have surface antibody on them, and they also have MHCs on them, right? So the antibody, the B cell is triggered when it matches antigen. It's identified as foreign. The B cell engulfs the antigen and digests it. The antigen is broken into little peptides in basically the same process that we've seen before. The little antigenic fragments get bound to its own MHC2 complex, bring it up to the cell surface. The cell surface, the MHC antigen complex on the top of on the mature the mature B cell, I can't speak this fast, will help bring and trigger some T helper cells to come to the site. And so these are the T helper cells here that have already seen this antigen as well. So they have that binding receptor here that is to the same antigen. There's the same antigen. B cell is bounded off to the surface. The T cell has it. The cytokines then can help the B cell to do what? To multiply and mature, proliferate and mature into those same antibody producing plasma cells that we've seen before, right? Just like we have over here. And we're also going to see they don't show it, I don't think. No, they don't show over here, but you'd also imagine that you're producing memory cells as well. These are, and then, so the plasma cells release the antibodies there, and they go looking for antigen. So those are kind of the two processes of B and T cells in the process. In both cases, it gets to the same endpoint of, from here down, they're the same process. This just is the T helper cell, and antigen presenting cell come in contact here, and in this case, the B cell is triggered already when it met an antigen. And it could have been this B cell. It could have been a naive B cell that develops through this process, this antigen-dependent phase, and then continues to go forward and meet more T helper cells to help with the process. As I said, unchallenged. I don't think I've said this. As I've said, antigen-dependent phase mature B cells can live for 6 weeks or more. Unchallenged B cells live just a few days. They're a very short life. Developing of plasma cells. We talked a lot about these in a broader context, but let's take a few minutes just to look at them and finish up here. So we've talked about them, that these are antibody producing cells. They're really large lymphs by comparison to a normal lymph. If you look at the cell size of a lymph, compare it to a red cell, there's a little bit of a mismatch, but they're usually about the same. Compare a plasma cell to a red cell, it's much bigger. The difference also is the nuclear cytoplasmic ratio. This has a very high nucleus to cytoplasmic ratio. This is about almost even. A lot of cytoplasm, but small, relatively small nucleus compared to the amount of cytoplasm. So the NC ratio is closer to equal here versus a regular mature lymphocyte. What do we see about this plasma cell? The next slide I think is a little bit better. This cell, this one, I think the other picture is a little bit prettier. We have this deeply basophilic cytoplasm that we see. It's really classic. When you see it in a slide, you'll know what it is. It's very, very deep blue. I used to say, if you have if you ever had the 64 count crayons, it's like a cornflower blue. It's a deep cornflower blue. Plasma cells are the ones that produce antibodies, IgM, and then others. We just talked about IgG. They produce IgA, IgE. Yeah, they're going to produce other antibodies, IgD. They're found in, not that we're not talking, notice it doesn't say peripheral blood. It says peripheral lymphoid organs. Those are the secondary lymphoid organs, as well as the bone marrow. They kind of hang out in the stroma of the bone marrow, where they can be under the action of some of the cytokines there. Comparatively, as I said, it's a much bigger cell. The immunoglobulins are in the cytoplasm, but very few of the plasma cells have those immunoglobulins that are hanging off the side, like we saw here, that they are surface immunoglobulins. These are surface immunoglobulins. That's not what we see in plasma cells. In plasma cells, we see the immunoglobulins developing within the cytoplasm. And you see these little vacuoles here. These contain the immunoglobulins. So they're producing immunoglobulins that form into little vacuoles within the plasma cell. Characteristically beyond the cytoplasm, color of the cytoplasm is the size, very big. The nucleus is pushed off to the side. They call it an eccentric nucleus. It's pushed off to the side. What else do we see? We see this clearing right here. This is called the perinuclear HOF, H-O-F, perinuclear HOF. It's a clearing. This is also the Golgi zone of the cell. So that's what this is. And then I, honest to God, in my wildest dreams would have never called this a clock face, but you really talk about the coarse chromatin structure of the nucleus. I think the other slide is a little better for this. See these chunks of nucleus? How it's very coarse and you can see, literally like little chunks, whereas this is a little bit more homogeneous. It's, kind of even clumping all across the whole thing. In a plasma cell, you get kind of clumpy nucleus. That's what this is supposed to be representing here, are clumps of nuclear material. Coarse chromatin pattern. They call it a clock face. If you ever hear someone say, oh, what cell has a clock face? They're trying to tell you that it's a plasma cell. I'm not so sure, but feel free to use that definition if it works well for you. Plasma cells are really, the most differentiated of the lymphocytes. It's really well defined at this point. It's past the point of just a mature lymphocyte. It has a very specific function of producing antibody to an antigen that's presented. We don't normally see these in the peripheral blood. You see them, as I said, in the peripheral lymphoid organs, in the bone marrow, the stromal cells of the bone marrow. And that's, the stromal cells and the stroma of the bone marrow is a really important feature of why the plasma cells hang out there. As I said, there's cytokines there that are going to aid in really continuous development of antibody by the plasma cell, as well as helping it to establish a pattern of longer life. So the cytokines there aid in developing a long-lived plasma cell. Normally, you're not going to see these out in the peripheral blood under normal circumstances. If you have a very high immune response, kind of a chronic immune response, you may see them. You may see them in another form where they're kind of halfway between a lymph and a plasma cell. You'll see them when you do hematology. They're called a reactive lymph. They're not quite a plasma cell yet, but they're clearly not normal lymphocytes anymore. We also see these, and I suspect that's the case of where we are now. We see them in some diseases that are characterized by very, very high protein in the blood, and things like Waldenstrom's macroglobulinemia or multiple myeloma. And the reason that I say I suspect that it's one of those here is because these red cells are really clumped together. And that's not clumping from another purpose. Yeah, these are just starting. It can be hard to see, but sometimes the protein in the blood can make the red cells look a little different. So there are different diseases where we can see plasma cells, but we're not going to normally see them kicking around. If you see one kicking around in a blood smear, though, don't freak out. It's, one of anything is usually okay. So that's the plasma cell, and that's its appearance, and that's its functionality. So really design, produce antibodies. That's what we're hoping for. Why do we need to identify lymphocytes beyond just this is a lymph? So we talked last week about how most of them are T cells, right? Why do we diagnose it? The book touched on this really briefly, a paragraph or two at the most, and we're going to talk about this when we talk about the diagnostic tools we use in the lab. It's called flow cytometry, where we're going to look at those cell surface markers. We're going to look at those antigens, those protein structures on top, the CD markers, and that the process of flow cytometry can help us identify and segregate each of those lymph populations into what kind of lymphs are they? Are they T cells? Are they B cells? Are they abnormal B cells? Are they, XYZ, kind of a lymphoproliferative disease? We can see all sorts of characteristics of these lymphocytes. A B cell isn't a B cell, isn't a B cell. They can have different structures and different processes within the host, whether it's kind of a normal B cell or it's some kind of a lymphoproliferative, more malignant kind of a lymph. So we're going to use, we use flow cytometry, and they use a process of using lab-created or lab-replicated monoclonal antibody that we'll talk about. Why do we do it? It really helps us pinpoint the malignancy of the host. If you know specifically what kind of a cell you're targeting, you can use a targeted therapy, and it's a little less of, to not put a kind of a gross analogy to it, but it's like, if you've ever heard the difference between, like a buckshot that is it's a gun pattern that, just spreads far and wide versus something really targeted. So in treatment for disease, you can try and treat, this kind of broad category of a lymphoid disorder, or you can find out what kind of lymphs you're dealing with and do some really, really close-in targeted therapy for treatment. And obviously, the tighter the therapy, the better, hopefully, the prognosis is. We use it to determine things like immunodeficiency or even immunocompetency of the host. We see it developed in HIV and AIDS. When they're looking at the bone marrow of the host, they'll look at it to see what kinds of cells there are. And just to be clear, flow cytometry doesn't only look at lymphocytes; it looks at any kind of cell that has those protein markers on it. So we'll talk more about that at another time. And last thing, I debated putting this on, and after I looked at it a second time, I probably wouldn't have done it, but I did, so let's just look at it really quickly. This looks at humoral versus cell-mediated immunity. Take this with a grain of salt here, people. I came from a site called thedifferencebetween.com, and I guess this is this is sort of my pet peeve one down here. Definition: adaptive immunity producing antibodies is humoral. Cell-mediated, cellular-toxic and T helper cells without the production of antibodies is the definition of cell-mediated immunity. B cell versus T cell. Humoral uses antibodies, and cell-mediated is cell-to-cell contact. This, I think, should say antigen detection, not antibody. So now I'm hating this table even more. Antigen detection is done by antigen. Antigen detection is done by antibodies. This doesn't make sense. And antigen detection by cell-mediated is done by receptors located on the cell surface. What's the mechanism? Antibody-mediated versus cell-mediated. Does it produce antibodies? Yes or no? what's its purpose, extracellular versus intracellular pathogens, immunologic surveillance. No, in the strictest terms, because remember, not until the B cell is introduced to the antigen will it be looking to do anything, right? So it's not like T cell immunity. It comes in first contact and does what? It can produce cytotoxic T cells that will kill immediately, right? This has to do some other process. And then this one I would not. This immunity against cancers, I think, is a misnomer. So I'm not crazy about that. I think what it does instead is can have some cytotoxic effect on tumor cells. So I think immunity is a strong word for that. And just to finalize, the humoral immune system, production of circulating antibodies, circulating proteins known as immunoglobulins or antibodies, means the same thing. Cells are B lymphocytes, which can differentiate further into plasma cells. Plasma cells produce and release immunoglobulins into the circulation, and B cells and plasma cells are important in the presence of prevention of primarily bacterial infections. And the rest is just the summary. So you can follow through that. There's the references, and we're done. As I said in my announcement, you can feel free to contact me on Tuesday if you want to try and meet for part of the normal lecture time. We're not having a lecture on Tuesday. We're going to have an online lecture. But if you want to come meet and talk about the test a little, if you have any specific questions, come see me. And we can either meet on Tuesday as a group for, a half hour or something and go over specific questions, or you can email me if you have specific questions. Okay? Have a good day. Bye.